Hello, welcome to everyone joining us for this month's ECR Wednesday webinar hosted by eLife, the series that aims to give early career researchers a platform to discuss issues important to you and your research career. My name's Emma, I work for the eLife Features team, and today we're sharing some advice for those of you who have recently started your first group leader position and those of you who hope to do so soon. The webinar will begin with the panelists sharing their stories. Then, in the second half of the webinar, we'll be putting your questions to them. To ask a question, you can type in the question box on the GoToWebinar Functions panel, or you can tweet us, we are eLife_careers, using the ECR Wednesday hashtag. Please also join us on Twitter immediately after the webinar, where we'll be continuing discussion under the ECR Wednesday hashtag. Finally, I'd just like to let you know that we are recording the webinar and will make it available on YouTube in the near future. Now I'll pass over to Babak to introduce himself and the panelists.
Hello, everyone. My name is Babak Momeni. I'm currently an assistant professor in the Department of Biology at Boston College. Welcome to ECR Wednesday webinar series. We have 4 panelists for today. Lilac Shiner, a group leader at Wellcome Trust Center for Molecular Parasitology. Scott Dixon, an assistant professor in the Department of Biology at Stanford, and Chris Hamill, an associate professor at Cold Spring Harbor Lab. Before we start, I'll give you a little bit of introduction about eLife and the webinar series. ECR webinar series are hosted by eLife, and eLife itself is a nonprofit initiative started by the support of 3 organizations, HHMI, Wellcome Trust, and the Max Planck Institute, with the goal of facilitating scientific discovery in life sciences. As part of this goal, eLife works towards accelerating the process of publication and at the same time trying to improve that process. For that, they have taken steps by using active sciences in all steps of decision making, making the review process more transparent and fair, and encouraging more open access publishing. I am also a member of the Early Career Advisory Group, or ECAG for short, at eLife. This is a group that eLife has put together, initially consisting of graduate students, postdocs, and junior faculty. And the purpose of this group is to act as a sounding board for eLife and provide eLife with ideas about what issues are important to early career researchers. It has been a great experience for me to exchange ideas with this group of talented scientists about issues that we all feel that are very important. If you're interested, I encourage you to get in touch and get involved in that process as well. The webinar series is an example of the outcomes that originated from ECAG. For today's webinar on advice for new faculty, I'll start by giving you my own perspective, and then I'll hand it over to so that we can listen to the advice of the other panelists as well. A little bit of background about me. I've had a bit of an unusual career path. My training and background has been in electrical engineering. Started at Sharif University of Technology in Iran, and then followed by my PhD work in electrical engineering and integrated optics at Georgia Tech. And I then transitioned by taking a postdoc position in biology and Show Lab at Fred Hutchinson Cancer Research Center with really very little background in biology. And I'm really grateful to them because they were willing to invest in my career and take me in. And then I started my current position at Boston College, and I've been really fortunate to land in a really inclusive and supportive department with great colleagues here. In the lab, you're interested in emergent properties of microbial communities, how interactions between different species lead to properties at the community level. And we use a combination of mathematical modeling and experimentation in tractable systems to answer some of the questions in the field at the intersection of ecology, microbiology, and systems biology. And the interdisciplinary nature of the work that we do has been a source of causing some challenges in setting up a new lab as well. Only my second year in this independent position, and in my experience so far, what I've found to be probably the most important factor is to stay organized. When you start a new position, it involves many new responsibilities, from coming up with new research directions to managing lab members and their activities, to applying for funding, to publishing, to forming collaborations. And each of these responsibilities have their own challenges, and it's fairly easy to get overwhelmed. Being organized, even to the point of being obsessively organized, is what I'm finding to be the way to stay sane. To give you an example, for maintaining the lab, we've taken steps and continue to take steps to stay organized. We keep a shared lab calendar. We have been using a database to keep track of strains and chemical inventory. And we keep the data and contributions from individual lab members in a shared drive so that everybody has access to them and they can exchange ideas more rapidly. And the process hasn't been perfect, but with the help and commitment of my lab members, it's been a major factor for coordinating lab activities and sort of removing some of the burden of setting up a new lab for me. The implementation is not complete. We are still in the process of organizing more, and we are in the process of transitioning to one of the collaborative platforms for exchanging ideas and documenting the progress of our projects right now. I'll stop here and turn to other panelists to hear their advice. But before that, there is a poll question for the audience, and I appreciate if you please participate. I think Emma can put the poll up now, please. Thank you. I can't see the results here, but I'm assuming that you're voting. And okay. I guess the majority are in the process of looking for a new position, and then followed by mostly junior faculty. I'll move to panelists now, and first we will hear from Lilek Scheiner from Wellcome Trust Center for Microbial Parasitology. Over to you, Lilek.
Hi. So I hope everybody can hear me well. Thanks, Babak, for the introduction, and thank you, eLife, for putting this together. I can say that it would have been very nice for me to have had an opportunity like that before I launched into starting a lab. I think those things are very helpful. So just a couple of words about my career track so far. So I think I will speak a lot about mobility. I'm originally from Israel, where I did my first degrees up until the end of my master's. Then I moved to Switzerland to do my PhD, and I moved to the US to do my postdoc. And now I'm in the sunny UK, Glasgow in the UK, at the Wellcome Trust Center for Molecular Parasitology, which is embedded at the University of Glasgow. So, yeah, I think an important aspect of my career progress have been being able to move around because of the sort of work that I do, which is molecular studies of parasites. I needed to go and seek for the right environments to get my training, and then for the optimal environment to start my lab. So it was very important to move around. And an aspect of that, which I strongly would recommend for any of the listeners, and I saw that the vast majority haven't started their lab yet, is to have support for so that your personal life are not affected by moving around, mainly from your family. So speak about those things, and make sure that you are making decisions that everybody in the family is happy with, because living in a new place could be challenging. So it's probably the best, when possible, is when you make that decision, you make it such that nobody feels like they're terribly compromising on their happiness and on their opportunities in life. And I've been lucky to be able to have that support and to make decisions in a way that everybody in the family is happy. And that's been super helpful, because starting your own lab, as Babak said, is a time-consuming, sometimes stressful, but even if not, then quite challenging endeavor. And so you really would like to avoid making that even harder by having your personal life suffer from that. I mean, it sounds like maybe more easy said than done, but in fact, I think that with good planning, it is possible. And I'll be happy to answer more questions about that later. The other thing where I will echo what Babak just said is that there is a lot to take over, and it could be overwhelming, or at least it's definitely very busy. And so it's true, being organized is very important, and prioritizing is super important. An advice that I got at the beginning, which I'm still following up, is to identify my productive hours and my less productive hours and my not at all productive hours, and dedicate those various hours to suitable tasks. So to give an example to what I mean, there is a lot of admin involved in running a lab, then there is people's management, and then there is writing of grants, of papers, and sometimes of emails that need a lot of sort of brain work. And for me, I know that brain work activity is the best early in the morning, or you know, at certain times of the day. And so those are the times where I would be writing grants or reading papers or writing papers, reviewing other people's grants, you know, reading people's theses and things like that. In the sort of middle level efficiency times, I would do things that are more to do with management, discussing things with the people who are working under my supervision, and trying to troubleshoot various things, either experimentally or administratively. And then in the least productive time is when I will try to do things that really don't need brain work. So to give an example, if I'm writing a review and I want to put together a figure, that's something that usually is time consuming but doesn't need a lot of brain work. So that's the sort of things I would do, you know, if I can't fall asleep in the middle of the night or something like that. Or you can think of some examples of that, those sorts of times. So I think that's been a golden advice that helped me a lot. And the other thing I would say about myself, again, to trigger relevant question, apart from moving around a lot, what I've done a lot in the past 3 years, so my lab is 3 years old, is writing grants, fellowships, and other funding applications. And I've been fortunate enough to be reasonably successful. I have a fellowship and a couple of grants, a couple of significant grants, and then a couple of small pots of money in my bag at the moment. And so if people have any questions about that, I'll be happy to answer questions like that as well. And I think that's all I have to say about me at the moment.
Thank you, Lilak, for sharing your perspective. Next is Scott Dixon, an assistant professor in the Department of Biology at Stanford. Scott, please.
Thank you, Babak, and thanks to everyone in the eLife team and the eLife side that put this together for organizing. I think this is great. I echo the thought that it would be something that I could have benefited from when I started my lab here in California 3 years ago. So I've been in this job 3 years. Before that, I was born and raised in Canada, did my PhD in Toronto, and that was in genetics. And then I switched fields from developmental genetics in worm to cancer chemical biology as a postdoc at Columbia University in New York City with Brent Stockwell. Then, based on that, I got the job in California. And so, speaking to the point that was just raised about being flexible with where you go, I think that the more flexibility geographically that you have or that you give yourself, the more options you'll have when you're searching out a career. Many people are constrained for various reasons to one place or one location, and that does make it more difficult. So I've been fortunate that I feel like I can travel, but it hasn't been easy. A point that hasn't been raised yet. I have 2 young children and a wife, and so moving the entire unit of people, first just myself and my wife to New York City, to a new country, and then the whole family of 4 people and cats to California, that was a big effort and definitely takes some planning and some adjustment. And there was a big adjustment when to a new place. Okay, so that's some of the background. What we do here is basic cell biology research. We do cancer cell death, so we're trying to understand pathways whereby cancer cells respond to different drugs and how they undergo apoptosis and other forms of cell death. So that's our main research interest, in being in a primarily research institution here at Stanford. That's been very helpful. We have access to clinicians and access to basic research scientists. But the more important thing I wanted to mention, I've been thinking about what I would say, and I guess I have 3 pieces of advice for all the people who are curious about new faculty. I would completely endorse and echo what has already been said about pieces of advice to follow. So I was thinking I would say one point that I've had to consider a lot is to be patient. When you're starting a lab, it literally is empty when you show up. There's nobody there. It's just you, and things are very slow getting going. Even if you're doing things yourself, you won't know necessarily where to find things, either physically in your lab space or administratively in the new institution where you're at. And so a degree of patience is required. Counterbalancing that, though, is persistence. So again, it is all you all of a sudden, and so nothing will happen unless you are the one that takes the lead on it. So you oftentimes find yourself, well, you have to rely on yourself quite a bit at the start, and so you can't expect anyone to do something for you unless you ask them. And that's maybe a difference even from a postdoc, where certain things would just happen. Your PI would be taking care of even simple things, and now you're responsible for all dimensions of the administration of your lab, as was mentioned. And so I think this applies obviously to the sort of constant worry of getting funding. You don't necessarily need to be persistent there, but also in every other domain of the lab. So for example, making sure that the protocols you develop are written down and recorded somewhere. It's one thing when you always would do that yourself as a postdoc, but then ensuring that your lab has the right culture to do those things that you want to be done, I think, is crucial. And so as a supervisor or as a PI, establishing the culture of your lab is your main responsibility. And that requires great persistence, because it's very easy, as Babak mentioned at the beginning, to set up a system where you organize everything in your lab, and I think that's wonderful. But to keep it going and maintain that requires constant vigilance. Then the last thing I wanted to mention was just that I think you realize very quickly this job demands a fair bit of flexibility along several different dimensions. So first, you probably arrive at the job with a set of ideas that you want to pursue, and you're trying them maybe for the first time in some cases, or eventually you'll reach the point where you're trying things that are completely new, and not all of them will work. So you have to be prepared, essentially, as some famous quotes along the lines of, make sure you have a plan, but understand that that plan won't necessarily survive its first encounter with reality. And so you have to be flexible in what it is that you choose to pursue. And I think part of that can be adapting your research program to the physical location you find yourself and the expertise of your colleagues and your institution. So if you move to a place that has a fantastic expertise in one specific domain, which often happens, maybe the domain is in proteins or in genetics or in some other aspect, that becomes something you should think about. And even if you didn't consider that when you were applying for the job, once you take it, there's no reason you wouldn't be able to shift your focus and take advantage of any resources that you have available to maximize the impact of your work. The other aspect of the flexibility, I think, would be, and something I certainly learned, is to take advantage of the expertise of the people you bring into the lab. So the graduate students you bring in, technicians, postdocs, they will come with much experience in science, which will be different from yours. And I certainly benefited from listening to their advice about equipment to purchase, ways to organize the lab, ways to run the lab, how to do specific experiments. You might have a certain way you think is best, but they'll be the ones doing the work. And if they have an equally good idea, then it makes a lot of sense to let them take the lead in doing that. And so if you can express that flexibility in various ways, I think that's really helpful, as opposed to trying to lay down one track where you think everything should happen. Certain things certainly should go the way you think, but many other things you can be more flexible. And then the final thought would be just to expect the unexpected. Many times, as a PI, you'll have a plan for the day, or you'll have your schedule that you want to follow, and then something will pop up that requires your immediate attention, and it's unexpected. I have a postdoc, and on the weekend, she had an accident and tore ligaments in her knee, and so now she's out of the lab. And that just happens, you know, and these sorts of things come up all the time, and there's not much you can do. But if you sort of approach it with a sense of, you know, persistence, I guess, and good humor, you can overcome these challenges. So I think those are the points I wanted to make, and I'll pass it back to Beth.
Thanks, Scott. Great advice. For our last panelist, Chris Hamill, I guess he's the most experienced panelist in this webinar. He's an associate professor at Cold Spring Harbor. Please, Chris.
Yeah, so I guess just from that quick poll, I realize I'm in the very minority section of that poll. So I've had my lab here at Cold Spring Harbor for just now 6 years. I just got promoted to associate professor recently. And I think, you know, after listening and not wanting to reiterate everything that the other two panelists have already pointed out, with two things really that I would say that I would expand upon what Scott said with building the culture of your lab. So now that I'm now in my 6th year, I'm sort of transitioning from the, you know, young hard Scrabble, just starting out, to like trying to basically have all the people together and me not doing very many of the experiments anymore, which is probably to me the most odd transition of this whole scientific career. So I'm used to seeing primary data and uninterpreted data, and I think that's one of the most complicated things because it requires, you know, personnel interaction, setup, and all that sort of thing. It's a hard skill to learn. I also, with regard to reiterating some of Scott's points about setting up the proper lab culture, I think in retrospect, what I would say is it was a sort of bizarre transition because I think I wasn't necessarily the 100% boss. And I look back now and say that I was a little too friendly, a little too senior postdoc-y when I first started my own lab. And that prevented me from sort of establishing this sort of, not that I want to say that I wanted to establish like ultra dominance of any sort, but it took away from my ability to move the trajectory, to accelerate certain areas, to motivate people in a lot of ways, to keep other people focused, which I would have thought that it would have been me that would have had the most hard time keeping focus just because I was divided enough. But it turns out that it's a specific task to get someone motivated when things aren't working and things like that. And being, you know, so I would say that if I look back, I would say I should have transitioned to being the more boss-like person much earlier on. We had a lot of fun. You know, we had a lot of lab outings where I felt, you know, in the mix and young and robust and everything now. But it is very important to me now that I'm the boss and sort of, you know, running the show and sort of making sure that the data that people are generating, you know, that I'm getting the uninterpreted sort of thing. So we're now moving into a lot more computational biology. And I find that's the hardest kind of data to really, it's not like a gel or a band or something you see and it's dark. I mean, when you have to go through all of the steps, the computation stuff, I mean, that's where I feel like I'm most vulnerable now for, you know, not being able to catch mistakes, which, you know, ultimately can cost someone a lot of time. So that's, you know, that's why I think being, you know, showing that you're the buck stops here, looking at all the stuff, that they have to present it to you in that way, was a hard transition for me to make. Secondly, I think the next point is that to learn how to get advice, and I think one of the things that maybe hasn't been said is that when you start your own lab, there's no shortage of advice. You get advice from people that you pass in the hall, you get advice from your, you know, graduate mentor, your postdoc mentor, and from my experience, the diversity of what they were telling me was so extreme that I found it sort of bewildering in a way. And so what I think that one of the most important very early on things to do is to get yourself associated with someone at your institution. So, you know, with regard to setting up, you know, ideas for grants, writing grants, reviewing your grants, and just stick with that person. And, you know, obviously make sure that they're giving you good advice. But I think sort of managing that sort of trajectory in the in-house sort of sense is an important thing to take advantage of early on. And I think that's pretty much it. I'll answer any questions that anybody else has. So. Yeah.
Thank you, Chris. Appreciate it. And I would like to thank all the panelists for sharing their experience and thoughts. And we now invite the audience to ask their questions, you may use the question box through the webinar platform, or send them to Twitter using the hashtag ECR Wednesday, and they will forward your questions to us. So the first question we've received is, how much did your projects vary from your postdoc labs, and how does this influence job prospects? I think you start with Lilek to answer that.
So that's a great question. I actually think this is, if you're now searching into moving to independence, this is really hitting the nail right on the head. I was fortunate enough to have 3 job offers, and I think a big part of that was that I was developing my own niche in the small field of parasite biology. That was a little challenging because the field is quite cozy, and that oftentimes happens that everybody does the same thing in waves, or not exactly the same, obviously, but very similar things. So this was an important step forward, and so I did start a new niche within our field, or at least I consider myself one of the people who did. And the way I did that was by taking expertise that I have learned in my postdoc lab, technical expertise, and applying them to a new research question. So I was still gaining a lot from my PhD and postdoctoral experiences, and sort of establish myself as the expert on parasite genetic manipulation, for example. And then I asked a new question. So in my case, I'm interested in the biology of endosymbiotic organelles. The parasite I look at have a plastid and a mitochondrion, and we're trying to understand how they are sustained. And so I started asking questions about mitochondrial biogenesis, which were not very heavily studied before in the field. And the way I also, in practice, established the fact that this is my own niche was to apply for an independent grant. I applied for, in the States, there is a grant called K99, which is a pathway to independent grant. And I put my idea in writing. I pitched it to a few people in my institute and got it thoroughly crucified. I went back and rewrote it, and then I sent it to this K99 scheme. And luckily enough, it got funded. And I think having an idea and also having some little formalized form of showing that this is that you own the idea was a very important aspect of getting job offers. So I think it's very good, whoever asked that question, that you're putting yourself in that position right now.
Thank you, Lilek. The second question is for perhaps for Chris. The question is, given the current funding climate, I am extremely concerned about the process of getting grants. How did you learn how to write grants? How much time do you spend writing grants?
Yeah, this is an example where I would say you need to learn how to write grants early on, and you need to have someone who has literally gone to the panels, because I think initially, so to sort of parlay from the last question, I sort of started a project when I arrived at Cold Spring Harbor that was very different for what I went and got the job doing. So I was doing something much more biochemical, and now I'm doing something much more developmental. So writing grants for that without very much preliminary data was difficult at the beginning, but what was important was learning how to actually mechanically write grants. And it is something that you need to get someone to sit down with you and tell you how to write grants. Because when you actually go onto these panels, you see the sort of unknown thing that you were just not aware of when you started writing grants as a young PI. So, and what I mean by that is that you are literally in your application trying to provide text that they will copy and put in the review of your grant. I mean, it is that simple. So it has to be extremely tight. It has to have the bold words in the right section. And this all sounds very simple, but it really requires someone, as Lilac said, to come in and trash your grant. And it will take something of that nature. And it seems, I think I see now, so now I'm sort of in the part where I'm helping the younger people out now. And it is a brutal process, but there is a formula. There is some sort of formula that you have to be successful. And I can look at my early grants, which are essentially proposing the same types and styles of experiments, and the funded grants, and they are slightly different, but I think it's really down to providing the kind of information that these people who are reviewing 10, 15, 20 grants need to memorize, put down in their notes, put down in the reviews. And I think learning that requires you to really pair up with someone that you can take their advice. So someone who can be brutal but being constructive as well. It's a hard thing for me now that I'm helping slightly younger people do it, to avoid being brutal, but there is a sort of pattern and system in order that needs to be in place. And it's very simple once you learn how to do it. So that's a skill that, there are also things like the Cold Spring Harbor lab here has outreach programs where people can come and learn this from panelists from NIH. You know, it's worth taking that sort of little miniature course. Just to know what goes on in the room is something that I think is an eye-opener. You know, in the panels for grants, for instance, R1 or NSF grants. I think I didn't have any idea what actually went on, and it seems insane what actually does go on. But writing toward that insanity and writing with the throughput that you need to be successful is something that I didn't appreciate early on. But yes, it is scary.
Please.
If I could just add on that specific advice, which I think is an excellent advice, to try to put yourself in the mode of the reviewers and the panelists. For those listeners who are in the UK system, I was just told very, very recently that you can ask to be an observer in certain of the UK research panels. So do that. Go and ask around your department, the people who are sitting on panels or your grant officers, and ask to do that. That is indeed extremely helpful.
Thank you. That's great advice. The next question is for Scott. They're asking, Scott, can you tell us how you balance being a new faculty member with being a parent?
Okay, that's an excellent question. And the answer, simply enough, is just with difficulty. Those two things are very much in opposition most of the time. So for example, to put it in practical terms, I work pretty much from when I drop my children off at daycare or school now until I have to go and pick them up. So I look on with envy at my other colleagues and peers who can, come in at 10 in the morning and work till 8 or 9 p.m. with a great amount of flexibility, then go to the gym and then go home and go to bed. So when you're a parent and a scientist, any of you in the same boat, you are balancing those two things constantly. And speaking to the flexibility aspect, definitely being a parent adds another big unknown element to that. Kids get sick, things happen at school. There's always something like school's closing because it's a parent or teacher development day, or there's holidays all the time. They have spring break. Next week it's grade 2, but they get a spring break here, so that's a week where they need to be taken care of. So when I was a postdoc, the daycare closed down for 10 days because they had mice in the building. There's just a whole host of things that really you wouldn't expect. So I think you can manage that partly by having colleagues in the same situation that you can commiserate with. That's been very important. If you're fortunate enough to live in your family, that's obviously a huge benefit. And when you're making the calculation about where you might search for a position, that has to enter the equation, I think, in some way. That if you're planning to have a family, where your family is or where your partner's family is a big deal. Now, we unfortunately, in some ways, we've moved as far away from all of our family as we could possibly get, so it's all on us. But then that means you have to try to access those resources that are available in your community, or, for example, here where I am, they have the school will provide emergency daycare for those crazy situations where things happen. I guess on the plus side, it does force you to be very efficient, as many people told me it would. That's turned out to be true. I don't tend to have much time to waste because I know that I have hard deadlines literally every day of the week where I have to leave the building. So meetings are scheduled and happen at those times. Work is completed by certain deadlines and so on. So I think there is some benefit there, but it's a huge challenge, and you're definitely slowed down compared to colleagues who have no family.
Thanks, Chris. Scott. And then next question is for Lilac. What were your startup packages? What can be expected for new faculty?
Sorry, I had to unmute myself. I think that is widely diverse, and maybe I would even invite each one of the panelists to answer. In my specific case, I had an offer as a senior lecturer in Hebrew University in Israel, which would have come with $500,000 as startup money for equipment, and because the lab would have literally been empty, and it would have come with my own office and with some level of commitment for PhD students over 5 years. It would have also come with a lot of teaching responsibilities. On the other hand, the offer here in the UK was quite different. There wasn't a massive startup package. Things were more in the vicinity of 200,000, and they included salaries. So you can imagine that for equipment, not much was left anymore. But on the other hand, I came to a department that already had a lot of parasitologists, so many things were available for me to sort of start using and hit the ground running until I got money and got my own stuff to expand the capacity of the facility. And so I didn't start on my own. I started immediately with a postdoc and with a technician, and that's quite a different type of start, and also with much less teaching responsibilities. So I think these things vary widely between countries and types of institutes. So I guess the important thing is to figure out what's the best for your career development and for the experiments that you plan to do and for the research that you plan to undertake, and apply in the places that will provide you those things, not so much in terms of what the money is, but rather what you can actually do with what you're getting.
Thanks. If Chris and Scott want to add, please.
Go ahead.
Please.
Okay, I'll go then. Going in order. I think the actual dollar amount, I think I signed something that's supposed to be confidential, to be honest, so I'm sorry. But I want to just want to echo what was said, that it's sort of, I won't say it doesn't matter, but it actually matters far less than you might imagine. For example, if you have, let's say you have $1 million, and you feel like you need to buy a certain piece of equipment that costs $500,000, and that's part of why you got your startup. Then your startup is really only $500,000, let's say, and another institution might offer you $500,000, but they might have that $500,000 piece of equipment next door, and you're allowed to use it as much as you want. Likewise with salaries for individuals, some places part of that is paid for by the university, and other places you have to cover it yourself, including your own. So really, a lot of it's actually an interaction between the dollar figure, maybe you might say, to put it bluntly, and where you are and what resources they would have there. So yes, it matters, and it is a thing, but I think actually it interacts with a lot of other variables that impact how much real value you get from it. And so the other thing, I did a spreadsheet at one point when I started, and the amount of money you need just to support salaries, including part of your own, let's say, and postdocs and technicians, over even 10 years is massive compared to the amount anyone would ever get in a reasonable startup package. So it really is just to start up the lab. It's not a career package, it's a startup package. So you need to get grants anyway.
Yeah, I would second that also, but I think when I was on the job market, so when I came to Cold Spring Harbor at the dollar value amount, the startup was far exceeding any of the other offers I had. It also happened to be the place that I wanted to end up, so it was a no-brainer. But one thing that I would say that you have to think about when you're looking at the offer letter too is, it turns out at some places, and I've had colleagues that this has happened to, your startup isn't forever. I mean, so you have to spend it. So like, I got a fairly large startup at Cold Spring Harbor, and it's unrestricted funds, and so I still have a lot of that money left. So that is money that I can use to move laterally at any time I want to. And in fact, I almost moved laterally within a year or two after coming here. And if I had spent all that money or hadn't had that unrestricted funds that could be extended out now into the 6th year, that would have been horrible. I mean, so like saving the startup money as flexible money, I think, so get money from grants as quickly as possible. That's obviously a good thing, because oftentimes the startup money is a lot more flexible than the money that you have for a particular grant. And I think that's something that I have leveraged now at least twice. That would have been very difficult to start a project completely from scratch with money that I had to get for that particular project. So that's something that you should check in the details. But I agree that the actual dollar amount is only meaningful when you compare what you have, what you need, and what you have to buy. I think that's it's different at different institutes. So that $1 million somewhere is not the same as $1 million somewhere else. So.
Thank you. The next question is again one of those questions that probably you can ask everyone, but we'll start with Scott. How did you adjust to teaching and getting research done in your early years?
Okay, I think so I'm in the Department of Biology at Stanford, which means we teach undergraduates. So teaching is a big portion of the job. And as is common in most places, you get protected time, usually your first year, and that's part of something you can negotiate. In the end, it didn't actually work out that way. I started teaching after a few months, but it was a graduate seminar course. So that was, and that was with another professor, Jessica Feldman. So that turned out to be very low input. Then in my second year, I developed a new course in chemical biology, and that certainly was more effort. But one of the reasons I wanted to come to this type of department was that I wanted to teach. I have 2 parents who are teachers. I grew up in that culture, and so that was something I was actually looking forward to. So it takes time. It definitely takes time away from your research. As my postdoc supervisor used to tell me when he was teaching his big biochemistry course at Columbia, that would take up mostly all of his time except for lab meeting during that semester. And I think that can be true if you want to do it well. So you have to budget that in, but I think for a lot of us, that's something we value. So we don't see it as a drawback necessarily, but as a great way to help the next generation and improve your own thinking about your work.
Thanks. Chris, if you want to add something, probably.
Yeah, I mean, so at Cold Spring Harbor, we don't have the undergrads, and I have to say I missed it. So I actually end up volunteering to teach at the Watson School, which is a graduate program here. And I agree with what Scott was saying. It is a great opportunity to think about something, from the primaries, and build something from the fundamentals on up. But from my perspective, I really enjoy teaching because it's my real opportunity to recruit graduate students. And that is, I have to say, if someone asks me in all honesty, I do it because I try to find the best graduate student in that particular class, and I want to recruit them to my lab. And that's, a necessity, I think, recruiting the right people to your lab, and again, reinforcing the idea of building the proper culture is important. But it does take time. So when I volunteered, I was thinking, I'll do this for X, Y, and Z, but it does, every weekend before a set of lectures, my weekend's destroyed. And even 3 or 4 years now into doing the same sort of developmental biology core course, still I have to get ready for every time. So it is something that is time consuming, but worth it in the end.
Well, I don't know if Lilek wants to add more, or should we go to the next question?
You could go to the next question, mainly because I benefit from very low teaching burden. And at the moment, for me, that works well. I teach graduate courses, which I think are a little easier to teach because they're all very, very close to what I do on a daily basis. So I think in that level, I probably don't have too much to offer.
Thanks. And the next question, again for Chris. Besides top papers and successful grant writing, what is very important for your CV? On the basis of what did you get your position?
Can you just repeat the question? Sorry, it was distorted sounding.
Sure. Besides top papers and successful grant writing, what is very important for your CV? On the basis of what did you get your position?
Okay, I catch it now. So yeah, I think one of the most important skills that is important even when you're a postdoc is your ability to socialize with people. And so, I mean, I just happen to be someone who really, really loves going to meetings. So that was part of the attraction for me coming to Cold Spring Harbor, because there's, you know, one every week. But I was the same way at whatever institute I was in. And so, like, I would say, you know, making yourself your own entity at meetings, you know, interacting with people, I think was very important for me to get the job here. In fact, you know, it was essential, I would say. Because I think in a lot of cases, when you go on the job market, a particular department, if you want to go there, they're not just going to pick your thing out of a stack of 200 applicants, even if, because the top there is the top everywhere. So you need to stick out. And so, you know, I invited myself to places that I wanted to get a job interview at, to give talks. I mean, I literally cold called people, you know, I mean, it's not, you know, it's not uncommon.
Thanks. And the next question for Lilek. Any advice on picking students or postdocs?
Yes, I echo, I think it was Chris who said, or maybe both Chris and Scott said, how important it is to have the right people. So I definitely echo that. I think everybody could imagine that that's the case. And having the right people is the combination of at least two things. One is how you as a supervisor and the student or postdoc or trainee of any level communicate. It's very important that you manage to communicate well. And the second thing is, what are the skills of the person you're taking? And so both of those things are things that you can assess before you take somebody in. And the thing I tend to do as much as I can, so in some cases you get to be lucky enough that there is a student who is around, either because they did a rotation project with you during their honors or master's degree, or because they have worked for somebody you know very well, or because you know them from the field. And so then you already know the person a little bit, and those are the best ways for you, because you've already interacted with them over a long period of time. But even if you don't have that, I would suggest to instigate that as much as possible. So what I tend to do when somebody contacts me to work for me at any level is, firstly, if they didn't already do that, then shoot back at them, why are they interested to work with me? And I would usually search for an interest in the science, and also to understand what their career aspirations are, so that I can know where they fit in with the sort of people I would like to recruit for different things. The next thing I would do after that is, if you know, if it's a satisfactory reply, then I will say a little bit about what's going on and what I would expect from somebody to do, and send a couple of papers or even a grant proposal or something like that, and then have them coming back to me again and explaining again the specifics of what interests them or what doesn't interest them in my proposal, and if they have a lot that doesn't interest them, then come up with some alternatives and explain themselves. And then I'd follow up with that. It sounds very time consuming, but I think it's worth it. I'll follow up with that with a meeting or two, one-on-one, either by Skype, or if the person is around the department, then one-on-one in my office. And again, usually bring up the same sort of questions, and just discuss them, you know, in a fluent way. And then things come up also about more specific, you know, you get the opportunity to trigger people on their broader understanding of the subject, or their general sort of expectations from their work over the long term. And then the last thing I do is make sure people want to work with us, meet the other people in the team. So my team is 6 people big now, and I try, if possible, that they interact at least with 1 or 2 people. And then firstly, that's good for them to know how I would be, and in some cases that might put people off. And I think that's important because, again, the matching is really important for both sides. And that also gives me then another opportunity to ask the people who spoke to the candidate what they thought about them, because the interaction with people at different levels is always very different. So those are the things I would do when I try to get somebody to join the lab.
Thanks. And one quick final question. This one is for Scott. The question is, I imagine early on you must have all had to triage some tasks that you just didn't have time for. What did you let drop, and how did you decide?
That's interesting. I guess, well, a couple things happen. One, what you triage is your like personal spare time, you know, like hobbies and things like that. I think I used to have some. They're gone. I don't know what they were. I have a family and I have the lab, and so you sort of push away a lot of stuff, you know, that you thought was essential to your well-being, and you realize it's just TV. And so you fill up more space with stuff to do. The second thing is when you're starting the lab, I think you have actually less than you'd imagine to do in the first year anyway. You're not necessarily teaching. There's some administration to do. You don't have really a lab. You're still getting equipment and people into the lab. So you're not necessarily dropping anything important, I guess. But I will say one story from when I was a PhD student, and if my PhD supervisor Peter's watching, you might not remember this, but I was labeling tubes in the lab next to him one day, and he said, why are you labeling tubes like that? You know, you could do it more efficiently if you just put, you know, short forms. And I said, you know, I'm just going to label them this way. And I think that is the sort of thing that definitely disappears once you've been a PI for a little while, is that you give up on those little tactical details and you focus on the big picture. And I think that's maybe where you do the most triage.
Was Peter Roy your advisor?
Yeah.
That's good. I would say, so one thing that maybe is not specific to this particular question that I noticed about having to manage multiple projects, because when you're a PI, even if you are working on the same subject, it's like you're trying to move 10 pieces forward at once, you know, depending on how many people you have in your lab and how many kind of independently running projects you have. One thing that I've realized after having a certain number of people is that you find that you're spending an inordinate amount of time with the slowest, most rate limiting person at the expense of a lot of times with someone who's creative, generating data, and so you have to learn how to manage that. And you know, my impulse at the beginning was, and this is where I was saying my sort of philosophy has changed, I wanted everyone to feel successful and have their projects and everybody to go home and like, I love my job. Sometimes people are just not that good, and sometimes they're just going to be inherently slower. And you can't dedicate all your time picking up the bottom. The consequence is the whole force doesn't move as quickly. And I think that's something that I spent a lot of time and a lot of anxiety over. Because basically once you get up to a certain size, and not that my lab is very large, but you realize that there's these kind of dynamics that you've never had to deal with before. You certainly didn't deal with them as a postdoc. But it's learning how to deal with those things that I think each person deals with differently. But it was something that was an emergent sort of property of running, 10 things that you wanted all of them to work magically. Sometimes things are just much slower, and sometimes this one person is much slower. So.
Thank you all for your contribution. So the questions can continue on Twitter for another hour after the webinar. And in the few minutes that's left, we'll go back to the panelists for sort of the closing remarks. If there is one take-home message that it's something that they would like to emphasize for everyone, I would invite them to share that with us. Starting from Lilac again, please.
I always have to start. I think take advice. Somebody mentioned that, I think it was Chris, specifically about grants. I think do that with everything. If there is a mentorship scheme, join it, find yourself a mentor who you like how they've done for their career. If there isn't one, force somebody to become your mentor, and take advice in every step. When you hire the first few people, when you write the first few grants, when you write your first paper, when you write your support letter for grants and papers, don't be shy. Firstly, people enjoy helping, and second, you really don't always predict all the things you should think about when you do those things. So do that as much as possible.
Yes. Okay. Well, thanks again for the organizers for this. I think it's been great. Not to end on too negative a note, but just one piece of advice maybe would be to be, again, persistent and be prepared for things to fail maybe early on, and understand that this happens for everyone. Your first grant probably will be rejected for various reasons, as Chris said. Your first independent project, maybe your ideas won't work, but you have to push through that, and I think most of us do, and we learn a lot about our own capacity, and I think that can be a beneficial thing. But it's tough at first, but it really is enjoyable. It's a fantastic job, and I wish everyone the best of luck at it.
Thanks, Scott. And Chris?
I would say basically this is you're at where you basically have been preparing for mentally for the longest time, and you're there, so you should learn how to enjoy it. It does come with a certain amount of anxiety. I mean, I can count very rarely the nights that I didn't think about something, or man, I really hope this works, or man, what if I don't get that grant, or there is a lot of anxiety, but you have to learn how to just go back and reflect on the fact that this is what you were dreaming about 4, 5, 6 years ago, was getting here. And it is an immense responsibility, but it is really actually fun on a daily basis. And if it's not, you should make it that way, because it can be. So.
Thank you all. So this is the end of our webinar on advice for new faculty. I'd like to thank again eLife and all our panelists for contributing, and our audience for participating. And again, if you would like to join the follow-up discussion, find us on Twitter at ECR Wednesday hashtag. Thank you, and enjoy the rest of your day.